It is my distinct honor to introduce you to the recipient of the 2021 AC Redfield Lifetime Achievement Award, Dr. Ronald Benner. This award honors major long-term achievements in the aquatic sciences, including research, education, and service to the scientific community and society. Ron Benner is the Chair Emeritus and Carolina Distinguished Professor at the University of South Carolina in the United States. Ron is receiving this award for transformative and enduring revelations regarding the origins, composition, and reactivity of organic matter in Earth's ecosystems, from inland waters to the deep sea. Dr. Benner's distinguished career in aquatic microbial ecology and biogeochemistry spans 3 decades. Beginning by uncovering the reactivity of plant lignin and terrigenous organic material in the ocean carbon cycle, Benner has made numerous and sustained contributions to our understanding of the origins, transformations, and fate of organic matter in the ocean. Among many discoveries, he is perhaps best known for his conception of the size reactivity continuum hypothesis, which describes the reactivity of organic carbon along a size gradient. Additionally, his work contributed to the development of the microbial carbon pump concept. Benner's full body of literature on these topics are highly cited, with over 25,000 citations. Dr. Benner has been an active educator, mentor, and member of the aquatic science community throughout his career. As an ASLO sustaining fellow, Benner has made numerous and varied contributions to the broader ASLO community, including through chairing conference sessions and serving on committees. Dr. Benner holds an Einstein Professorship with the Chinese Academy of Sciences for advancing scientific collaboration between the United States and China, fellowships with the American Geophysical Union and the American Association for the Advancement of Science, and many other distinguished fellowships and appointments. Nomination letters reflect on characteristics of Dr. Benner's approach to science. They say that Ron does not tackle easy problems. He takes a broad perspective on these problems and then focuses on asking insightful fundamental questions. He approaches experiments with elegant simplicity that also leave no room for ambiguity. Regarding the combined impact of Dr. Benner's contributions, they note that his research on dissolved organic matter has fundamentally transformed our understanding of the global carbon cycle. Dr. Benner's contributions have been enormous, spanning limnology, coastal and blue water oceanography, ecology, and organic geochemistry. In addition to impacting current knowledge, Ron's findings have ramifications for understanding how the biosphere will respond to climate change over the next few decades. Please join me in welcoming and congratulating Dr. Ron Benner for receiving ASLO's AC Redfield Lifetime Achievement Award.
Amina, I'm not sure.
If you switch to presentation mode, you'll be perfect.
Okay, thank you. All right, well, this is a very nice introduction. Thank you, Amina, for the kind words and introduction. And I'd also like to thank Bopi Badanda for nominating me for this award, and to thank David Kirchman, Marilyn Fogel, Farouk Azam, and Jim Kottner for letters of support. Most of my career has focused on the origins and transformations of aquatic matter and of organic matter in aquatic ecosystems. I had an early start on that in the salt marshes on Sapelo Island, and from there eventually moved to the coastal ocean and to the open ocean. And today I want to just highlight a few of the research accomplishments during those years, and in the end try to tie things together with a bit of some insights and maybe some speculation about regulation of the carbon cycle. This beautiful academic family tree was put together by Aaron Fichot. I greatly appreciate it. I'd like to start off with just noting that Bob Hodson, my PhD advisor at the University of Georgia, Bob was always very supportive, encouraging, full of good advice, and a great friend. So I really thank him for all of his efforts with me as a graduate student. And in this family tree, academic family tree, in the lighter blue, the names are students who worked in my laboratories at the University of Texas and the University of South Carolina. The darker blue are postdoctoral associates. Thanks to all of you. You all contributed greatly to shaping the research that we did, and I appreciate everything you did. You helped make the whole journey through research a lot more fun and made it a big adventure. So I was fortunate early in my career to meet Marilyn Fogel and John Hedges and start collaborations with them. Marilyn introduced me to stable isotopes, and John introduced me to organic biomarkers. I've really been fortunate to have many collaborators throughout my career. There's a long list, but to all of them, certainly my career was greatly impacted by all of you, and I appreciate the spirit of collaboration and sharing that we had. Overall, I would say that these collaborations showed me the value of combining experimental and geochemical approaches in research. And throughout my career, I've sought to use both approaches to understand the origins and transformations of organic matter and to integrate those observations over space and time. So my doctoral research was on plant litter decomposition, and I was focused on the major structural biochemical components of plant litter, which would be polysaccharides like cellulose and hemicellulose and lignin. These components typically comprise 50 to 75% of plant biomass. The major observations coming from my doctoral research were that the polysaccharide components are typically degraded at rates that are 2 to 4 times higher than rates of lignin degradation. Under aerobic conditions, the rates of biodegradation are about 10 times higher than they are under anaerobic conditions. And probably the most significant and novel observation from my doctoral studies was that lignin is actually biodegradable in the absence of oxygen. Another important finding was that in aquatic systems, bacteria play a major role in lignin degradation, whereas in terrestrial systems, fungi seem to play a larger role than bacteria in lignin degradation. And finally, one of the observations from my doctoral research was that during plant litter decomposition, dissolved forms of polysaccharides and lignins are released into the DOM reservoir, and that certainly got my interest in dissolved organic matter, which I ended up spending a lot of my career working on. So in collaboration with Marilyn Fogel, we had a project that we wanted to look at the major biochemical components like the cellulose, hemicellulose, and lignin components of plant tissues and look at their stable carbon isotopic composition. So we isolated these various fractions, and Marilyn measured the carbon isotopic compositions of them. And one of the things that we found consistent across a broad range of types of vegetation was that the lignin component is depleted in C13 by 4 to 6 per mil relative to cellulose and hemicellulose. That's a large isotopic difference, and it was an exciting discovery, and at the same time, from our knowledge of rates of biodegradation of these various components, it suggested to us that the bulk isotopic composition of decaying plant material might change during decomposition. So we had a long-term 18-month study of the decomposition of Spartina root and rhizome material that was buried in litter bags in the sediments. And over the 18 months, we saw cellulose and hemicellulose removal about 55%, quite a bit less removal of lignin, which is consistent with previous work we had done, with only about 30% of the lignin removed. But with those changes and the knowledge that the isotopic composition of lignin is quite depleted in C13 relative to cellulose and hemicellulose, it suggested that the isotopic composition of the material would be changing over time. And in fact, when Marilyn analyzed the isotopic composition of the litter bags samples, what she found was that as the concentration of lignin increased in the decaying plant material over time, the isotopic composition was depleted. And over the 18-month period, after about 55% mass loss, the isotopic composition was about 1 per mil, shown at the top there for the 18-month samples, over 1 per mil difference. So that's significant, and it influences how we interpret bulk isotopic compositions. When we developed techniques for isolating and concentrating lignin from dissolved organic matter, whether it's in fresh water or salt water, it provided us the opportunity to characterize lignin phenols and quantify them in all aquatic systems. And so I was very interested in this question at the top: what happens to terrigenous dissolved organic matter in the ocean? And that question could be addressed with lignin phenols, because lignin phenols are biomarkers. They're only produced by vascular plants, and therefore the source of lignin phenols to the oceans is predominantly through riverine discharge to the oceans. So by measuring the concentrations, composition of these lignin phenols, what we found in the Pacific Ocean was very low concentrations and yields of lignin phenols, and in the Atlantic, very low as well, but substantially higher than in the Pacific. So in the Atlantic, the concentrations were 2.6 times higher than those in the Pacific. And that's consistent with the fact that there's 3.6 times greater riverine discharge to the Atlantic. So that looks very consistent with riverine discharge as the major sources as well. We also measured bulk stable carbon isotopic composition of these DOC samples, and we found that they were similar in the Atlantic and the Pacific. And that indicates that they have mostly a marine origin of the DOC. And that's consistent with these very low concentrations of lignin phenols, which are indicative of Terrigenous DOC. The other thing, looking at the discharge to the basins and the volume of the basins, we could come up with some residence times for these lignin phenols and terrigenous dissolved organic matter in the ocean basins. And in both basins, it's relatively short. So these short residence times, the low concentrations, they both suggest that there must be substantial removal of terrigenous DOC in estuaries and ocean margins. And that's something we went on to pursue in a variety of systems, but it really culminated with a major study of the transformations and fates of Terrigenous DOC from the Atchafalaya, Mississippi River system in the northern Gulf of Mexico. So the shelf area is shown in this figure that we looked at. So it's a very large area that we covered the entire ocean margin. We had 50 stations, and this had 5 expeditions over a 14-month period. And this slide just summarizes all of the work that was done in this study. During the study period, there were 2.63 teragrams of Terrigenous DOC coming from predominantly the Mississippi Atchafalaya River systems onto the shelf and margin, mostly the Louisiana shelf. Over the span of the study, 0.68 teragrams of that TDOC was mineralized in the mixed layer, shown in the yellowish color in the figure. And then another 0.29 teragrams was below the mixed layer, but above the picnic line. And that's where that space in the surface ocean covered 85% of all the shelf fresh water. So you can see that a large fraction of the Terrigenous DOC was mineralized on the shelf, approximately 40%. The residence time on the margin there ranged from about 60 to 90 days for that region. The major mechanism of removal is shown here as biomineralized DOC. So this is microbial degradation. Another important mechanism is photo-enhanced biomineralization. So photochemical alteration enhances biological utilization. And so that's shown here. And then also photochemistry directly can mineralize the organic matter to CO2. That was a much smaller fraction, but it was significant. So that's shown as well. So in fact, what we observed with the low concentrations and short residence times that we saw for the open ocean, this study and many others have now demonstrated very clearly that a lot of the material coming in from rivers is decomposed and removed in coastal waters and on ocean margins. The size reactivity continuum of major bioelements in the ocean was something that kind of evolved over time. And one of the first pieces of information that really sort of got my attention is when we developed methods of tangential flow ultrafiltration for trying to isolate dissolved organic matter from the ocean. and also from rivers, freshwater environments. It separates not primarily by size. And so when we looked in the ocean, we found that most of the dissolved organic carbon was in this low molecular weight fraction. In fact, if you look at the top diagram, which is total organic carbon in the oceans, this is the size. So this is nanometer size. This is micrometer size. Actually, particulate organic carbon and particulate carbon only accounts for a very small percentage, meaning above 0.7 microns, only a few percent of the total organic carbon in the ocean. And that includes all biomass as well. So by far, most of the carbon in the ocean is in this dissolved phase and colloidal phase, down to 1 nanometer in size is the cutoff between high molecular weight and low molecular weight, which is shown here. So by far, most 80% of the TOC in the ocean is actually low molecular weight, and that is really small, less than 1 kilodalton, and that means most of the organic carbon in the ocean is non-living, and it resides in these very small dissolved molecules, which was the first clue, because we normally think of about the size reactivity continuum, because we normally think of small molecules as being readily accessible to microbes for utilization. So we wanted to look at the utilization question directly, and did some bioassay experiments where we took seawater samples, separated the high molecular weight DOM fraction from the low molecular weight DOM fraction, and did incubation experiments with natural seawater inocula and these two forms. And in fact, what we saw very quickly was that the high molecular weight DOM is more rapidly utilized. So this is dissolved oxygen. So we're looking at respiration is much higher with high molecular weight than low molecular weight DOM. We also looked at microbial growth, found the same thing, higher with high molecular weight DOM. And here is DOC removal for high molecular weight DOM and low molecular weight DOM. So in every case, bacterial respiration, growth, and DOC removal were considerably higher with the high molecular weight DOM than they were with low molecular weight DOM. We also were doing chemical analyses of different size fractions of carbon that we could recover from the ocean. And we did chemical characterizations of them. And we found just as there's a size reactivity relationship, there's also a size and chemical composition relationship. And that's shown in these various figures. In this figure, top A, if you look at plankton, this is just plankton toes at the Hawaii Ocean Time Series Station. And this is characteristic of many other samples that we've analyzed. Amino acids comprise a very large fraction of the biomass, as do carbohydrates like neutral sugars and amino sugars. So about 40 to 50% of the biomass is just as those few common biochemicals that make up proteins and saccharides, polysaccharides. So that's in plankton. And then if you look at POM, you see the fraction, and this is including living and non-living material. In the surface ocean, it's less. The deep ocean, it's even less than it was in the surface. High molecular weight, there's less common biochemical material in that than there was in particulate. And it goes down with depth as well. And finally, if you look at the low molecular weight DOM, both in the surface and the deep ocean, only 2-3% of the total carbon can be characterized as these common biochemicals. So there's a huge spectrum in the size, between size and chemical composition in organic matter in the ocean as well. This is also with sediment trap samples, you see the same situation. So putting that and other information that we had from a variety of different approaches together into this size reactivity continuum, we can see the following things, that actually the size of particles and material in the oceans is really driven by these two major biological processes. Decomposition, which is decreasing the size, and then biosynthesis, which is, of course, forming large particles through plankton and other organisms. And we also have aggregation and disaggregation, but these biological processes by far are the major ones for shaping both size. We also saw they shape chemical composition. As we get to these very small molecules, common biochemicals make up a surprisingly small fraction of the carbon. So there's a lot of uncertainty or complexity about what is the rest of this from a molecular point of view. Carbon, most of it is not known what its chemical composition structure is. And we also found that the smaller DOM is of greater age, as well as decreasing size and decreasing reactivity. So most of you, I think everyone is probably familiar with the major role that bacteria play in the microbial loop. And that was well characterized 30 years ago or more through a variety of studies showing that bacteria are the major decomposers and consumers of dissolved organic matter in the ocean. And in that process, they regenerate nutrients that are critical in the surface ocean. So it stimulates primary production. But we weren't thinking at that time of bacteria as actually being a primary source of the dissolved organic matter in the ocean, and in particular of what we refer to as refractory DOM in the ocean, which is just persistent to the, it persists in the ocean for periods of years, centuries to millennia. Some simple bioassay experiments conducted by Hiroshi Ogawa really made this very clear, in which very simple substrates were added to a seawater inoculum with glucose or glutamate in these two examples. And the glucose is very rapidly consumed, just as glutamate is. Within 48 hours, we can no longer measure the concentrations of glucose or glutamate. However, the concentration of DOC, it falls precipitously, but not down to zero. And you can see that it drops off, and there's typically 10 to 20 micromolar DOC, whether it's glutamate or glucose as the carbon source. And you can also see there's DON that persists as well in these. These incubations went on for over a year. I don't show all that data, but this DOC persists for more than a year. And its persistence indicates that bacteria could be a very important source, actually, of the dissolved organic matter in the ocean. We did some other incubations to really look more deeply at the chemical composition and structure of molecules that are being produced by bacteria in these types of incubation experiments. We did very simple substrate incubations or more complicated substrates called oligomer substrates that we added. And we also just compared them with a sample of coastal dissolved organic matter. And we looked at, with ultra high resolution mass spectrometry, molecular formulas and their elemental compositions. And shown here, you can see that the elemental compositions of the molecules, whether it's the monomer or oligomer substrate or coastal DOM, it's amazingly similar. So these were incubation experiments that went on for about 4 weeks, and yet they have very similar molecular occurrences as the coastal DOM with regards to elemental composition. If you look at the total molecular formulas, it's also not as big in the bacterial incubations compared to coastal DOM. However, it's amazing because that's the only thing we had in these incubations were bacteria to produce this. Coastal DOM, of course, has a lot of riverine inputs and a lot of molecules like lignin that come from plants. So overall, if we looked at how many of the molecular formulas in the bacterial DOM that we find in coastal DOM, it's a whopping 50 to 60%. So that was pretty interesting with regards to how much contribution there might be from bacteria to DOM in the ocean. And an even closer look at the structural aspects of this DOM is with two-dimensional NMR. And this is just kind of like, you don't have to look too much in detail at these. This is a surface Atlantic dissolved organic matter. And these are the two experimental monomer and oligomer. And just by glancing by eye, you can see that, of course, there's a lot more structural characteristics in the surface Atlantic than what we see in the bacterial DOM, but all components are basically there in every one of these. And that includes a group which we refer to as carboxyl-rich alicyclic molecules, or CRAM. We found in working with Norbert Hertkorn that this CRAM component of dissolved organic carbon in the ocean is large. It's on the, we estimate about 8% of the DOC resides in these kinds of molecules. And the bacterial DOM has those types of structures in it. So they are a source of CRAM in the ocean. Also, other studies now have been done in freshwater systems, including lakes, and very similar results in those samples as well. So an independent way to kind of look at how much of the dissolved organic matter or particulate organic matter in seawater is derived from biomarkers is not to do any type of incubation experiments, but to use biomarkers. In this case, we're looking at the D forms of D enantiomers of amino acids, and another unique compound found in the cell wall of bacteria, muramic acid. We're using these as biomarkers and just collecting samples from the ocean, analyzing them, comparing them with cultures, and then coming up with an estimate of how much of the carbon and nitrogen actually in dissolved and particulate carbon in the ocean comes from bacteria. In the top panels, we have the percentage of POC. This is again from the Hawaii Ocean Time Series station in the Pacific Ocean. About 30% of the POC, and 20 to 30% of the DOC is of bacterial origin based on these biomarkers. And in the particulate nitrogen, it's more on the order of 50 to 60% for particulate organic nitrogen, and again, 50 to 60% for particulate organic nitrogen from bacteria. So these studies, which are using very different approaches, are suggesting the same thing, and that is bacteria actually are a major source of the dissolved organic carbon and nitrogen, as well as particulate organic carbon and nitrogen in the ocean. So all of these studies led to the formulation of what's referred to now as the microbial carbon pump. And it's referred to as a carbon pump, and as shown in the diagram, you can see that microbes, and here showing bacteria, viruses, archaea, they are major consumers in the microbial loop of the detrital material, both in dissolved and particulate form from plankton. They decompose it, and some of the regenerated nutrients through the microbial loop are reassimilated by phytoplankton and zooplankton. But these microbes also release forms of dissolved organic matter that are resistant to decomposition or recalcitrant forms of DOC to the ocean. And that, once it's below in the surface ocean and into the deep ocean, that carbon can persist on timescales of centuries to millennia. And that's why it's referred to as a carbon pump. It's sequestering carbon in the ocean. So it's playing an important role in regulating climate. And so that leads me to the last part of my presentation, and that is trying to address what is certainly, I think, one of the major issues that humanity faces right now, and that is climate change. And how can this reservoir of refractory DOC in the ocean, what role does it play in the climate change scenario? This amount of carbon, which is shown here, 640 petagrams of carbon, is comparable to the atmospheric reservoir of carbon dioxide. So it's a very large reservoir. And small changes in the size of that reservoir, like a 10% removal back to CO2, would have a big impact on atmospheric CO2 and climate change. So up or down with this value in response to climate change is a big possibility. The balance between the production of DOC and the production of refractory DOC in the ocean and the removal of DOC in the ocean, the balance between those controls this reservoir size. And so what I want to look at now, since the work we've done over the past few decades on microbial decomposition of organic matter and removal of dissolved and particulate forms of carbon from the ocean, we want to investigate what controls that removal from the ocean. Again, we used bioassay type approaches, and these were long-term experiments lasting more than a year, and quite a few different experiments that accumulated to look at the removal of some refractory DOC that we collected from the ocean and then subjected to in incubation experiments to a variety of different conditions. Priming is when we add a labile carbon substrate that a lot of microbes can readily use as a carbon and energy source. Coastal microbes is just one example of taking a microbial population from one system and putting it into a different system. So these seawater samples that we collected this refractory DOC from was a ocean system, and we replaced, in this case, we added some coastal microbes. And then photodegradation, we just exposed it to a solar simulator for simulated sunlight to see if we could photomineralize and photoenhance the biodegradation. And those values are shown here with samples from the surface, mesopelagic and deep ocean. And you see removal is particularly large by the addition of coastal microbes and also by photodegradation and photoenhanced degradation. So there's some significant removal by these processes and by using microbes from a different environment and adding substrates that are very labile and readily used by microbes. So it gives us some insights about what factors and processes can remove this refractory DOC. And to summarize all of those studies, this figure is showing these various processes such as photochemistry, which can only, of course, happen in the surface ocean, typically only in the upper 50 meters of the ocean, where you have ultraviolet light penetration and absorption, alteration of molecules, some CO2 being produced, and labile substrates being produced for utilization by surface ocean microbes. Microbes in various regions of the ocean, the community structure and diversity varies quite a bit. It varies on small scales as well as large scales, which we're trying to show by this figure. But on the large scale variations, this would be very different type of population here in the mesopelagic zone or deep ocean zones. We have priming, sinking particles release labile forms of DOC, and that could cause this priming, so utilization there. In the deep ocean hydrothermal vent systems, it's been shown that they take in and they actually combust some of the dissolved organic matter, so they mineralize it as well. So one thing about all of these different processes and microbes, environmental conditions, is that the one thing that connects them all is circulation. Ocean circulation, we think, plays a very important role in shaping the removal of refractory DOC from the ocean. In particular, global overturning circulation, so the entire ocean water turns over on timescales of 1 to 2 millennia, and it gets back up to the surface. So what goes down eventually comes back up, it gets exposed to sunlight. It moves microbes, it moves molecules into very different environments, and mixing through global ocean circulation, we speculate, would enhance the removal of refractory DOC from the ocean. A number of studies done in the past decade have clearly shown that global overturning circulation is actually now slowing down with climate change. And what we would expect, if this is correct, if this speculation is correct, that is when we slow down global ocean circulation, we might be able to retain more refractory DOC in the ocean. And so that would be a potential negative feedback to climate warming. But that is, of course, very speculative. But I think that we need clearly a lot more studies to really understand what is controlling this very large reservoir. It's one of the largest reservoirs, surface active reservoirs of reduced forms of carbon that sequester carbon from the atmosphere. So we really do need to know what are the controls on it. And with that, I think I will finish up, and happy to have some discussion and questions.
Thank you so very much for the wonderful presentation. Everyone can join me in giving a virtual clap to Dr. Benner. And we don't have time for questions right now, but questions in the chat we'll definitely send to Dr. Benner. So thank you so very much. Congratulations on this wonderful award. And now I will turn the space over to Isabel.